Dear students, so in the last lecture we talked about the microbial ecology in terrestrial systems, and today we are going to talk about microbial ecology in aquatic systems. Usually it is assumed when we hear the word aquatic we are talking about surface water, so we might be referring to ponds, to lakes, to rivers and other surface water bodies. However, it's interesting and important to note that aquatic ecosystems are not limited to surface water systems. In fact, the water can exist in air, land, like on the surface of land and below the surface of land. And thus today in this class we'll be talking about microbes and the microbial ecology in context of how do they come there, how do they survive in these ecosystems, in air, on surface and below the surface. So let's get started. Okay, so here we have the three different ecosystems, one for atmospheric water, surface water and groundwater, which is below the surface water. So let's go through them one by one and then I will show you some specific pictures and schematics for each of these. Atmospheric water, in atmosphere, water is present, whether it is cloud, whether it is the water vapor, the absolute or relative humidity around us. This water might at times serve as a small habitat for microorganisms, and this is the reason why we have airborne illnesses such as tuberculosis and flu, many flu viruses that can be transmitted through these air particles, and if the air particles have the right condition, the right humidity, the right temperature, then they can persist in this aerosols for a long time. Now the aerosols, so they are not dissolved in the air, definitely they are suspended in the air. These particulate matter, these aerosols might be the good habitat for microbes to sustain, and in that case we refer to them as bioaerosols. Definitely bio means life in the aerosols. The next very important example of bioaerosol is the mixed-phased clouds. So in clouds where we have good amount of humidity, where we generally tend to have particulate matter, and definitely we have air, so we have a mixed phase system, the bacteria or any other microbe might find it a very suitable place to live in the cloud and then rain down upon wherever they rain down, in case they do, or just disperse back in atmosphere. It's very interesting that this atmospheric transportation of pollutants, including microbiological pollutants, is recently discovered phenomena, in sense that earlier we did not give much importance to it, but now we are realizing the very important relevance of airborne transportation of pathogens and microbes. A very clear example perhaps would be the example of flu. So every year we have a flu season in our side of the world, which is Indian subcontinent and Chinese subcontinent. And nearly after 6 months we have the flu season in the North American and South American continent. And it's interesting how the flu virus that is spreading the annual flu in our part of the world shares genetic similarity with the flu virus that will spread or would have spread flu 6 months later in the North America and South American continent, implying that there is a high probability and likelihood of the flu virus getting transported through some airborne mechanism from the Indian and Chinese subcontinent all over to Americas. And the other way is also true, that the flu virus from North and South America travels to India and China and actually across the globe, but we happen to be diametrically opposite, so it's a good example suggesting that the air microbiome is very important. And in fact I might say I want to take a segue and talk about how relevant this air microbiome is nowadays, is by the immense amount of research that is focusing itself on the air microbiome. We are very interested to see how the microbes are spread through the air, how diseases are spread through air, and how our indoor air is not only different in terms of its physical and chemical characteristics compared to the outdoor ambient air, but also in its microbiological perspective. So the one way to think about this is the moment we enter a room, we shed off microbes from our body. The moment a dog enters the room, let's say I have a pet dog and he enters the room, he's shedding off microbes all over the room. Now these microbes will travel to different parts of the room undergo transformation. Some will die, some will proliferate, and thus we'll have a very different air microbiome inside compared to outside. So now coming back to the atmospheric water, why would it be very important for us? Why we as human beings would be very interested in atmospheric water and the microbes that live in it? Well, obviously we are interested about public health, and it so happens that there are certain pathogens that do very well in these bioaerosols given the right humidity and right condition, and we'll talk about them. The other thing is very important because there are microbes present in the air, we call it air microbiome. I'll like to write this down actually. So we can split it into different parts. This particular word can be split. So definitely air is, air. Micro on a small scale. So this is giving you an idea of the size we are talking about. Bio means life. And when we talk about ohm, like omics, we're talking about a technique or a strategy that involves all the forms of biology present. So if you remember in one of the previous lectures, I talked about how less than or nearly 1% of microbes we know how to culture them, and we can culture them in lab, but majority of them are not yet culturable in our laboratories, and thus we go for high throughput amplicon sequencing based techniques which allow us to look at the entire microbial structure. Now this word ohm is a general suffix to many words, implying that everything is included. So air microbiome means all the life that exists in micro scale in the air. Obviously it is not possible to have a clump of microbes floating in the air, because due to their weight they will come down due to gravity. So we have definitely a size limitation. Now because the microbes are present in the air, so let's think of it this way. Let's say this is a bacteria, and let's say it's a clump of few bacteria, and there is some moisture around them, and they're floating happily in the air. And let's say the air has some organic contaminants or organic carbon, and they have some organic forms of nitrogen, and maybe not even necessarily organic, it can be inorganic form of carbon as well. So these potential electron donors, electron acceptors, nutrients and carbon source can very well be transformed in the bioaerosols. So if you remember we talked about the microbial energetics, and we talked about how microbes harness the energetic exchanges in their environment to use it as energy source, to use it as carbon and nutrition source. The same thing happens in the air. So this is very important because this might be actually catalyzing conversion of many primary pollutants into secondary pollutants. So again, a short revision from your basic environmental science course, it's taught to all undergraduates now. Primary pollutant is a pollutant that is originally released from the source. So if I have a chimney, it's releasing some smoke from the industry, that's a primary pollutant, all the pollutants in the smoke. Now when it goes out in the air, it comes in contact with light, comes in contact with other phenomena including bioaerosols, and then it undergoes a transformation and makes secondary pollutants. Alrighty, many a times the secondary pollutants are more harmful for public health than the primary pollutants. So we are very concerned about it, and we are particularly interested in knowing how bioaerosols play a role, and we are finding out increasingly they play a good amount of role. The other thing is bioaerosols and air microbiome in itself is very important, because remember how we teachers taught you for many years how clouds are formed. We told you, well, the nucleation happens in such a way, there's a particle and then the water condenses around it, and then it grows, and then lo and behold it's a mini droplet, which then becomes a droplet, and then next thing we have is raindrops, right? So it does not always have to be a particulate matter, an inert particulate matter. In fact, it can be bacteria or a group of bacteria that are sticking to each other. So bacteria can also serve the role of particulate for nucleation of rain and ice. And this is very important. Let's say we talk about ice nucleation. So when we talk about ice nucleation, not only are we talking about how microbes will help or are continuing to help formation of clouds, formation of rain, but also if the temperatures are low enough, then we might have preservation of these microbes for a very long time. Let's say there's a bacteria or there's a virus or there's any other kind of microbe that's pathogenic, and we are not interested in ensuring the persistence of that microbe in the environment, but it undergoes ice nucleation and now it has a protective layer around it, yeah, which maintains its temperature to be low, and this can be transported over long distances. So apart from affecting the weather, the local weather conditions and the local chemical scenario, so what kind of pollutants you have, what kind of chemistry you have, and what kind of pathogens you have, what else do air microbes do? As I mentioned about seasonal flu, this is very important. The particulate matter travels all across the globe. So pollutants from, let's say, released in China will cause acid rain in Tokyo and will cause Los Angeles to suffer excessive deposition of dust. So it's not just particulate matter that's getting transported, but it's also the microbiome. So we are increasingly interconnected. We always have been interconnected in terms of bacteria. Alrighty. So now let's move to surface water. So this was atmospheric water, and we'll briefly go over it again. In surface water, well, one of the biggest thing about surface water is that we are very concerned about public health. And why would that be so? Because from large part of our world, yet definitely our country also, surface water is still the primary source of raw drinking water. So we take surface water like a river water, lake water, we treat it and then we use it as drinking water. Now if the quality of our surface water is not good, then we are in trouble. So when we talk about surface water and surface water microbiology, ecosystems, we are very interested in what its repercussion would be for public health. So we are interested in pathogens, and continuing with our interest in public health, the next thing is algal toxins. So most of you must have heard about eutrophication, where we have excess of nutrients and lo and behold things grow and our lake dies, our water body suffers, and at times we also have algal blooms, and some algae, not all, some algae produce algal toxins which are not good for us. We'll briefly go over that again. And then coming back to the basic parameters which affect the persistence of pathogens, which affect and can drive the algal growth of biofilms, are BOD, organic nitrogen, and other nutrients. So let's look at what BOD is. I have briefly mentioned about BOD in past. I think it's a very good time for me to talk about BOD. So BOD stands for biological oxygen demand. Some people also call it biochemical oxygen demand. Basically it is the amount of oxygen that will be consumed in 5 days or in any t amount of time, depending on what my time duration for BOD is. And why would this oxygen consumption happen? Because we assume and we believe that there are organics present in the water and there are microbes that want to eat it. So if organics are present in water and I provide microbes that love to eat it, I want to know how much the oxygen would dip in t amount of time. T could be 3 days, T could be 5 days, T could be infinity, depending on what I'm interested in. So the more the BOD, in other words, the more oxygen is consumed, we know that there were more waste to begin with. So I like to think of this in terms of, let's take your favorite food. Let's say your favorite food is noodle. You love noodle, let's say. Maybe you don't, but let's imagine, or pick any other food you like, okay? Idli is one of my favorites, so we can pick idli or anything, okay? Really, pizza. So you take, let's say you have 1 kilo of noodles. You can finish them. You can finish them. If you're a bacteria, you can finish them, let's say, in 1 day. But now I increase the load of the food and make it 100 kilos. Now the 100 kilos will take longer time for you to finish alone. But let's say you can't afford longer time. We have limited the time. So from 0 to 3 days or 0 to 5 days or 0 to infinity, whatever, we have limited the time and we make enough humans. We bring enough humans in to finish the 100 kilos of food. Now the oxygen that would be consumed by the humans or by the bacteria to consume the 100 times more amount of food will be directly proportional to amount of food. So if there is less food for bacteria to digest, they will need less oxygen. Okay, so let's remember, for redox reaction there are two components to it. One is the reduction and the other is oxidation. So if we have lot of food, and food here is both carbon source and food is also the electron donor. typically food, when we talk about food for bacteria, it's biology, it's organic waste, and they are all electron donors. So if I want to consume electron donor, so if I want electron donor to undergo oxidation so that it can release energy for microbes, something has to be reduced. So what will be reduced? Electron acceptor. And in case of biological oxygen demand or biochemical oxygen demand, however we put it, that electron acceptor of choice is oxygen. That's why it is oxygen demand. So we don't know how much waste is present. We don't know how much electron donor is present. But what we can, and we can't measure it. We want to find out how to measure it. So one way we can measure electron donor is we can find out how much oxygen would be consumed to finish this food. Or in other words, we can find out how much oxygen would be consumed by microbes if there are enough microbes to eat as much electron donors as they can within time period. So let's say in first 5 days, let's say so I take water sample, 2 water samples, sample 1 and sample 2, and in sample 1 I make sure that both samples I make sure that there's enough oxygen, and after 5 days, and I add bacteria by the way, so I take sample 1 and sample 2, okay, both have enough oxygen, both have some amount of waste material. I don't know how much biodegradable waste material or how much electron donors they have, so I will add lot of bacteria to them, or the bacteria that I know will eat up these in case they are not already present, will eat up the electron donor. So now bacteria will try to drive this reaction. So let's say after 5 days of incubation, I noticed that the dissolved oxygen in sample 1 has dropped by 5 milligram per liter, and in the other it has dropped by 3 milligram per liter. So this is the delta in DO. This is the consumption of dissolved oxygen. So I know that more oxygen was consumed in sample 1. So if more oxygen was consumed in sample 1, I can say more electron donor was there to be digested with this oxygen. More of it was present. So I can say that the more electron donor was present in sample 1 compared to sample 2. In other words, more waste material was present in sample 1 compared to sample 2. So this is what BOD is. By looking at how much oxygen is demanded, I get an idea of how much organic or biodegradable waste is present in the water. So this is what we call as biological or biochemical oxygen demand. Alrighty, so in surface water, this is one of the most important parameter that we love to measure, because just by looking at BOD, we sort of know how fast microbes will grow. Because let's say in the beginning I don't have any microbe present in the water, but I have a very high BOD loading. Let's say I have 700 milligram per liter, which is pretty high. Actually, it's pretty high, yeah. So I have 700 milligram per liter of BOD, and there are no microbes. But eventually, because there is food, the microbes will find a way, and once they find a way, they'll proliferate. So I know the potential of how much the total microbial growth can happen in this. I know how much heterotrophic plate count I can expect from this sample pretty quickly if it has such high BOD loading. So BOD loading tells me an idea of how much microbes can grow. So this is very important. And the other thing is, the question is, where did such high amount of organic waste or biodegradable waste come from? So probably it came from a sewage, raw sewage, or partially treated or incompletely treated sewage. So if it's coming from sewage, then I can definitely expect fecal contamination, and there's a very high chance of pathogens being present. So high BOD also tells me that there's a high chances of pathogens being present. The other thing is, if it came from industrial sources, not sewage. Let's say there's some industry that has very high BOD load, okay? Let's say biscuit industry, and they have lot of it will probably be higher than 700 milligram per liter, but they have a very high BOD load, high amount of waste that they're releasing in their effluent, and there are no pathogens present, there are no microbes, but eventually over time they're very likely to consume it. The other thing is, if it is industrial waste, there's a very high chance that they have other chemicals that they're also releasing. So let's not take biscuit, because we assume that everything in biscuit is edible and healthy or not unhealthy. Let's take pharmaceutical industry, and they have very high amount of BOD, right? Or let's say considerably high amount of BOD. So they are not just releasing biodegradable waste of food for bacteria, which bacteria will eventually consume and bring it quite below to the nearly detection limit, or below our regulatory standards, but they are also releasing other contaminants that are not good for us. For example, they might be releasing a lot of antibiotics, Yeah, so they might be releasing lot of antibiotics, the pharma industry, or they might be releasing lot of endocrine disruptors, which are basically chemicals that hurt us, hurt our endocrine system, and thus upset our hormonal cycles. So whenever there's a high BOD, our alarm should go red, and the red herring should tell us either there are a lot of pathogens or a lot of contaminants present, or there's a huge potential for pathogens to grow. Alrighty, so again, when it comes to surface water, we are very interested in BOD. The other thing we are interested when it comes to surface water is how the electron gradient, how the light gradient and energy yield changes with depth. Alrighty, so let's take case of lake. Now remember in the previous lecture when I talked about population, communities and guilds, I drew a diagram of a lake. So I encourage you to go back and revisit the previous lecture and note that as we went below from the surface of the lake and we increased the depth thereby, the energy yield of any redox reaction reduced, the electron acceptor gradient went from aerobic reactions to anoxic reactions, which included nitrate reduction, ferric reduction, sulfate reduction, sulfur reduction, all the way to methanogenesis, and then to all the way to fermentation and then methanogenesis and acetogenesis. So we notice that the microbial communities in surface water will vary with depth. So one kind of microbes might be very prevalent in the top surface, may not be so prevalent in the deep surface, and this is where a lot of research is going on, and we'll briefly go over how microbes change through depth. Then obviously temperature is important. The lakes or relatively stationary bodies of surface water, they usually have a temperature gradient and undergo twice a year or once a year mixing. So this is mixing would happen when the top layer and bottom layer have such temperatures that the bottom layer wants to come up and top layer wants to go down. Now we know this, that water is most dense when it is at 4 degrees Celsius. Anything less, it's lighter, anything more, it is lighter. So again, as the temperature of the atmosphere changes, temperature of the top surface changes, the lake will undergo mixing cycle, and you will study this in other courses, environmental science courses, or in general EVS course also. So as the mixing happens or does not happen, the microbial communities also change. The other thing with surface water that we need to be careful about, so first thing we need to be aware of is depth. The other we need to be aware of is the electron acceptor gradient, and then we need to know about definitely how deep the light goes. Because of turbidity, the depth might change. Then we need to know about the mixing, how mixing is going on, and the next thing we need to know when it comes to surface water is how the nutrients are changing with depth. So nitrogen levels are changing and the phosphorus levels are changing. So we will study through about these things in subsequent slides. Now another important thing about surface water is that, in fact, whether it is atmospheric water, surface water or groundwater, if they are clean, if they have not suffered heavy human impact or impact of human activities, and they are relatively clean, they are likely to have less amount of nutrients compared to other environments. For example, some parts of the lake might be oligotrophic, some parts may not be. For example, the sediment zone may not be lacking essential nutrients, but wherever the water is clearer and cleaner, it might lack the nutrients, and in that case the oligotrophic microbes, they devise certain techniques to survive. One of them is motility. They learn how to swim towards more food. So the bacteria that you notice or the microbes you notice growing in surface water bodies, like even ocean or lake, pond, river, would have beautiful whip-like structures that help them float, that help them move, whether it's a flagellum or any other extrusion. So one is motility, that's how they develop. The other is that they make biofilms, and this is the last point in the slide for surface water, biofilms and motility. So in biofilms, what these microbes do is that they stick with each other, and I think in one of the previous in the upcoming lecture when we talk about drinking water microbiome, I'll be telling you more deeply about biofilms, what biofilms are, how they are helpful. Briefly for now, it's important for you to know that when microbes stick together, they are better able to capture the nutrients in the water, so they act like a sieve and they capture whatever nutrients go. They also protect each other, so it's almost the penguin effect. You know how penguins club together and when a storm comes, and only the penguins at the periphery of their group, of their heap, will suffer, and the young ones inside are saved from the storm. So this is exactly what the bacteria do. And in biofilms they have an added advantage that they secrete extra polymeric substances. They secrete them and they make matrix around them, which gives them further protection. Alrighty, then we'll move on to groundwater. Again, if not surface water, then our next source of water is groundwater. People have been thinking of getting water from atmosphere, but the technology still needs some more development for mass utilization. But groundwater definitely is a major source of raw drinking water. So our first concern again is public health. Now when we think of ground, definitely in the first few feet, up to the first few feet of the below the earth's surface, we will see lot of nutrition, and we will see a lot of microbial communities, whether they are anoxic or facultative aerobes, facultative anaerobes, or aerobic microbes. In the reason is because this is very close to the surface. It gets a lot of nutrient deposits like biomass that deposits on it, and then human activities impacted, and that's why it's not unexpected to see quite rich microbial communities and diverse microbial communities in the top surface of the soil. Interesting thing happens when we go deep towards the deep aquifers. So initially it was an assumption that with groundwater microbial growth is not an issue, but now we are noticing it is. Not necessarily in terms of severe pathogenic presence. There are two pathogens, we'll talk about them, but there are microbes in and around groundwater who are very happy to be in those oligotrophic, even oligotrophic, sometimes not necessarily oligotrophic, but definitely anoxic zones and in presence of water, and they really not only affect the quality of water, but they also affect the minerals and the chemistry of the soil around them. We'll talk about this. And so let's see here. So this is biogeochemistry. So bio, life, geo, earth, chemistry. So the life in earth, it changes earth and it changes earth's chemistry. The other thing about groundwater we are interested in is contaminants. So often what happens is we spill things, and then the spill percolates down and reaches the groundwater, and our groundwater is contaminated. Here I have the word NAPL, non-aqueous phase liquid. So these are non-aqueous, so non-water phase liquid. So when they reach the groundwater, they have their own missile, they have their own plume that they make. They don't necessarily mix in the groundwater. These are one kind of contaminants, especially petrochemicals. They will definitely make a plume, a NAPL plume. Now depending on the physical and chemical characteristics of the plume, it will undergo degradation, it will undergo mixing to some extent, and transportation through the groundwater, and affect the microbiology. I have drawn this diagram before, but please go ahead and take another look at this. Alrighty students, so take a look at this diagram. Let's imagine that this is the surface of the earth, and there was a tanker carrying oil, and it spilled the oil percolated down the earth through the layers of the terra, and here we have groundwater. Now please note that this diagram is just a schematic, just a cartoon of groundwater. Groundwater does not flow in waves like this, it just passes through the earth's pore. So it looks very different, but it's quite a discernible and significant flow. And let's say it's flowing in this direction, so from my right to left. And this is when the oil has spilled and percolated down. Because it is non-aqueous, it will not mix with the water as much, and because of the surface tension of water and the contaminant, let's say it's petrochemical, they will try to form a missile, a blob of NAPL, non-aqueous phase liquid. Alrighty, over time and pretty instantly also actually. The napple will undergo slight dissolution, so some of it will have a slight equilibrium with the water, so this is the red thing, and as it moves ahead it will undergo a shape deformation, like this. So definitely this is an exaggeration or may not be an exaggeration. So an apple is here and the dissolved level looks like this, and the reason for this is the resistance provided by the earth or the soil pores to movement of napple. And when this happens, because before this happens also, when the dissolution happens, the microbes start eating whatever they can, because you know microbes, bacteria and other microbes, they love to live in, they're hydrophilic, they love to live in water, and that's how their cellular membrane is made. If they live in oily places, then the cellular membrane will be disrupted. Remember, hydrophilic and hydrophobic ends. So the cellular membrane makes use of the fact that inside the cell and outside the cell it's a water based medium, and the integrity of the cell membrane is maintained. So they don't like to enter into napple. If they do, they won't persist very long, most of them at least, but in the dissolution layer they will grow, and as they grow they will start consuming the electron acceptors. They start consuming the napple, which is electron donor, and the more they consume, the more of it would dissolve, and after some time the volume or the amount of napple left would reduce. Yeah, the volume total might increase, but CV, concentration into volume, would definitely reduce with biodegradation. Now as remember, this is electron donor, so some electron acceptor is also getting consumed. So the first electron acceptor to get consumed will be oxygen, energetically most favorable. So at the periphery of the dissolved plume we still have some oxygen left, so this will be your aerobic zone, and then you will have anoxic zone. Now in anoxic zone you might see nitrate reduction, you might see iron reduction or sulfur reduction, sulfate reduction, and so on and so forth. And we'll notice that the closer we come to the plume, we'll go to anaerobic metabolism, which will look like fermentation or methanogenesis. Alrighty, or acetogenesis. And the reason is that the first dissolution layer will happen here, right, very close to napple, and the microbes will consume it right away. They're waiting here. When the dissolution starts, we start consuming. So they will deplete the oxygen here first, near the napple, and then eventually when that is consumed and the napple has traveled further apart, then here the oxygen will be consumed. Further apart, here the oxygen will be consumed. For now it has reached here, so this is the aerobic the aerobic layer where oxygen is still left. So we notice that in case of groundwater, we might, in the same local region, we might get very different microbes. So here we might have aerobic microbial community, here we have nitrate reducers, sulfate reducers, methanogens. And thus groundwater can be very diverse, both in terms of function and in terms of phylogeny. Alrighty. So in groundwater, the oligotrophic environment plus the contaminant create pretty interesting phenomena, and we notice that we have very diverse microbes. So notice all of them tend to have very diverse microbiology. Atmospheric water is very important for us because of bioaerosols and how diseases spread geographically. Surface water, because still the raw drinking major source of raw drinking water, and groundwater because in many places we have contaminated it and it affects the biogeochemistry. So we are very interested in understanding the microbial ecology of groundwater. Now the other thing about groundwater is that usually they are bacteriologically safe. But there are two pathogens that are known to survive groundwater conditions. One of them is E. coli and the other is Cryptosporidium. Now keep this in mind and remember it for when we are going to talk about drinking water microbiome. Alrighty, dear students, I think this is all for today, and in the next lecture we'll go ahead and we'll talk about atmospheric water, surface water and groundwater more in depth. So I will show you some of the cycles that undergo in all these three ecosystems environment, and how they impact public health and why they're important for us. Thank you.